Okay, so this question we are going to discuss, which is from GATE 2021 Life Sciences paper, and from Biochemistry section, Life Sciences Biochemistry section, question number 18. So the question states, with respect to sodium dodecyl sulfate polyacrylamide gel electrophoresis, so which is basically SDS page, which of these statements is or are true? Ethidium bromide is used to track the progress of electrophoretic mobility. Beta-mercaptoethanol is used to reduce disulfide bonds. The protein migrates towards the anode, and the lower molecular weight protein migrates slower than the larger molecular weight protein. Okay, very important to note here that this is a multiple select question or MSQ. So you will have multiple answers to this. That is point 1. The second point, which is the most important point, is that some of the MSQs will have negative marking and some MSQs do not have negative marking. So that you have to check in the question paper whether the particular MSQ that you are solving does have negative marking. Discuss some bit about SDS page first. Okay, so this is the SDS page system, which is sodium dodecyl sulfate and polyacrylamide gel electrophoresis. So polyacrylamide is one polymer which actually makes a matrix or a gel kind of when we mix acrylamide and bisacrylamide together. In a particular ratio. So usually the ratio is 29:1, which makes 30% polyacrylamide. So this is a very usual ratio that we use normally. We can also make different other ratios of acrylamide and bisacrylamide, and so that depends on which are the samples that we want to run in that particular gel. Okay, so this is a mix of acrylamide and bisacrylamide. So acrylamide itself is a long chain, so it is itself a polymer. But why do we need bisacrylamide? So acrylamide is a long chain kind of a structure, and bisacrylamide is used for linking those chains. So it forms branches in those chains. So together that forms a gel matrix, which is called the polyacrylamide. Also remember that polyacrylamide is a neurotoxin, so it has to be handled with care. Whenever we are using it in our labs, we should always wear gloves while handling polyacrylamide. So this is the polymer that forms the gel. So what we do in this particular setup is that we run proteins. Usually we run proteins, or we can also run DNA in these gels. So this is how the gel looks like. And it has these veils. So we have a structure like this. This is called a comb. Which we insert into the gel, so that these kind of wells form in the gel. These are the wells where we can load our sample. So you can see this here, that with a pipette, with a tip, we can load the sample inside the wells of the gel. And this is a vertical gel electrophoresis system, so it runs in a vertical direction. And along with the proteins, we also use something like this, which is called a protein marker. So what does it do? What function does it have? Exactly. So it is used for comparison of molecular weight. Very good. Of the protein. So this say SDS page is a system where we resolve proteins according to its molecular weight. So obviously we need a standard, like suppose our protein like this, for example, here these proteins are, here you can see that the proteins have formed different bands. So which protein is of which molecular weight, that we can determine from this standard, which is called a protein marker. And then what we do is we run these samples under electric voltage. So we apply electric voltage, and these proteins, these are all negatively charged proteins. How these proteins are negatively charged, we are coming to that, but these proteins are all negatively charged. So you can see that the proteins will move towards the positive electrode, which is an anode. So how do we remember that anode is positive and cathode is negative? I usually use this method, that anions move towards the anode and cations move towards cathode. So since cations move towards cathode, so that means cathode should be negative, and anions moving towards anode, so obviously anode should be positive. This you remember, and never forget anode is positive and cathode is negative, because often we face this confusion while, especially while in exams, we are kind of confused which one is which. So don't confuse, remember this formula or this thing, that cations always move towards cathode. So obviously if cations are moving towards something, that should be negative. So that is why cathode is negative and anode is positive. All right. Now coming to this, that the proteins we apply electric voltage, and the negatively charged proteins, they move towards this positively charged electrode, which is the anode. Now while moving towards the positively charged electrodes, the proteins have different molecular weights. So different molecular weights means that some proteins are larger and some proteins are smaller. So the smaller proteins, they will face less resistance, so of course they will move faster through the polyacrylamide gel. So therefore the smaller proteins move faster, so they come towards the bottom of the gel, whereas the larger proteins, they face more resistance while migrating in the gel, so therefore they remain in the upper portions of the gel, so they migrate slower. So that is how we resolve the proteins according to their molecular weight. Now another question is, where if two proteins are of same molecular weight, then we will not be able to resolve them, because this technique is completely based on this information, that what is the molecular weight of the protein, right? Because it's a separation completely based on molecular weight. All right, and then after that what we do is we stain the gel. So there's a dye which is called a Kumasi dye, so which is used to stain the proteins, and then we place the gel in this dye solution, and after some time we take out the gel and then we de-stain it. So there is a process of staining and de-staining, after which we will be able to see something like this. So our marker will be visible, along with our protein bands will also be visible, and we will be able to determine the molecular weight of our protein of interest. Now going into more details, how the proteins are negatively charged. So here comes this important factor, which is SDS. SDS is sodium dodecyl sulfate. Okay, so what it does is that it imparts negative charge to the protein. Now before going to that, I will also like to discuss the role of 2-mercaptoethanol, or which is also called beta-mercaptoethanol. We often write it as beta Me. Okay, beta-mercaptoethanol. So what it does is that it reduces the disulfide bonds. So usually the proteins are in this form, like the proteins are in their native state. Suppose we have purified a protein and the protein remains in its native or three-dimensional state which is a folded form of the protein, where there can be disulfide bonds, there can be positive, negative, different kind of charges on the proteins depending on the amino acid residues, right? And so they are folded proteins. Now we cannot, I mean we can, obviously we can run these kind of proteins on page on a polyacrylamide gel, but that will hinder the process of separation of the proteins according to their molecular weight. Why am I saying so? Because when the proteins are folded, they will be separated according to their shape and not their molecular weight specifically, because a shape of the protein and molecular weight of the protein can be very distinct. I mean, even if the protein is of low molecular weight, it can have a shape which hinders its migration in the gel, so it will migrate slower comparatively. So that is why if we run the proteins as it is in their 3D form in the gel, we will not be able to separate it according to their molecular weight. But that is also done, and that is called native page, or we also call it non-denaturing page. So where the proteins are not separated according to their molecular weight, but the proteins run in their 3D form, in their 3D structure. However, in SDS page we do not do that. SDS page is a denaturing type of page, and first of all we treat the samples with beta-ME or 2-mercaptoethanol. So what it does is that it reduces the disulfide bonds. So these disulfide bonds are broken by beta-mercaptoethanol. And then we also treat the protein sample with SDS. So what it does is that it first of all unfolds the protein. So this unfolded structure, it forms this kind of unfolded, so protein becomes linear, which is actually the primary structure of the protein. The protein becomes linear, and it also imparts negative charge which is proportional to the polypeptide chain length. So which means that the protein which has a longer chain length, I mean the protein which is a larger protein, suppose there are two proteins, one has 200 amino acids and another has 300 amino acids. So the SDS page, SDS what it does is that it imparts negative charge proportional to the polypeptide chain length, or which is also called it imparts uniform negative charge, which means that it will impart negative charge proportional to the chain length of the protein, which means that 300 amino acid long protein will have more negative charge, and the 200 amino acid protein will have less negative charge. Okay, because simply the chain length is longer in 300 amino acid protein, so it will have more negative charge. So which you can see here that this particular chain which is shorter, it has less negative charge, whereas this particular chain which is longer, it has more negative charge to it. Now how does it help? So if you have more negative charge, it will migrate faster, it will try to migrate faster, right? But the important thing is that this migration will be dependent on the molecular weight of the protein, because this long chain will face more resistance. Okay, so therefore the important thing is that due to more resistance, this long chain will migrate slower, and the shorter chains, they will face less resistance, so they will migrate faster. So this, for example, corresponds to the longer chain, this band that is formed in this gel, whereas these two, they form bands here, so they migrate faster. Okay, so this is the role of SDS. Now coming to the gel system. So in the polyacrylamide gel, in SDS page for it specifically, so there are two layers of the gel. One is called the stacking gel, another is called the running gel or the resolving gel. Any idea what is the difference between a stacking gel and the resolving gel? Right, yes, the pH is different. So in stacking gel the pH is 6.8, whereas in the resolving gel the pH is 8.8. Why such differences we are making? Because anyway the proteins are going to be running through the whole gel. So this difference is because in the stacking gel we want the proteins to be stacked in a particular line. In a line, it should form a line in the stacking gel. So how does it form a line? So we use glycine for that. So in the stacking gel there is glycine. So glycine has a pI which is close to 6.8. okay, so because of that what happens, this glycine is approximately neutral in this pH. So glycine forms a front, okay, and below that are the proteins and below that are the chloride ions. So the chloride ions are very small, okay, so we have chloride ions in it which comes from the tris HCl buffer, okay, and the chloride ions come at the front because they are very small, and so they form a front here. So for example there are these chloride ions, you can see. Then this forms a front, then there will be the proteins, all the proteins will be there. They will come in a line because after the proteins the front will be formed by glycine. So there will be glycine stacking on the proteins. So which is why in the front there are these chloride ions, on top of it there are these glycine molecules. So of course the proteins will be stacked in between them, and that is specifically what we want, the proteins to be all at the same line, because now we know that we will be starting to resolve the proteins, and if the proteins do not come to the same front then we won't be able to resolve them according to their molecular weight. It's almost like a race where all the runners are standing in the same line, they form a same line. If it's a linear format of the race, they all stand in the same line. So it's like that, the proteins all have to stand in the same line. So that's one thing, that's the role of the stacking gel. Then in the resolving gel, as you mentioned, that in the resolving gel the pore sizes are smaller, and there the proteins will be resolved according to their molecular weight. So the bigger proteins will be on the upper side of the gel, the smaller proteins will come migrate much faster. And the positive, there are these electrodes, the negative electrode and the positive electrode, and the proteins, they are all negatively charged because of the SGS that is attached to them. So now the negatively charged proteins will move towards the positively charged electrode. And how do we actually make this gel? So there are these two glass plates. So in the glass plates have a very small thickness in between them. So we attach them together, we attach them with a clip together, and then we pour the liquid gel in between them. And two important things we used for polymerizing the gel, I should have mentioned it before. Any idea what are the components of the gel? No, glycine comes from the buffer. So I will mention here also. the buffer is called TGS. What are the components? Tris HCl, glycine and SDS. Okay, it's called a TGS buffer. And so glycine comes from the buffer. Glycine we do not put in the gel itself. And in the components of the gel, obviously the first thing is polyacrylamide. The next is the buffer. So in the stacking gel we put the pH of 6.8. We put Tris HCl of pH 6.8 in the stacking gel, and in the resolving gel we put Tris HCl of pH 8.8. So we use two different buffers. Alright, next is, there are two other components which help the polyacrylamide to form a polymer, or for the gel to form, like gel to solidify. What are the two components? That is called APS. Exactly. Very good. APS and TMAID, these are the two important, very important components of the polyacrylamide gel. So what is the role of APS? APS initiates the formation of free radicals, and TMAID actually accelerates that process. So if you do not add these two components, your gel won't solidify. So it will be in a liquid form as it is, because like polyacrylamide, when we mix the acrylamide and bisacrylamide together, they are usually in liquid form. I mean, if you have powder also, you can dissolve them in water, and that will be your polyacrylamide, which will have 29% acrylamide, 1% of bisacrylamide, but that's a liquid. It will only solidify when we add APS and TMAID. So these are the two major components which is very crucial for the solidification of the gel. Now what is the total system? The total system is that you have this gel cassette where you have formed your gel, and you also have this electrode chamber. So you put this gel cassette inside the electrode chamber. You then you put this electrode chamber inside this electrophoresis tank. So there's a tank, big tank, where you put the gel and inside this chamber, and then you add running buffer. So I've already mentioned the running buffer is TGS, Tris, glycine and SDS. And so there are these electrodes, the cathode and the anode, right? And here what you do, after putting this whole setup in the buffer, then you add this sample. So this can be different types of sample. It can be a cell lysate, it can be a purified protein, it can be anything. So then you run you put the samples in those wells, right? And then you attach these electrode leads, the wires, to the power source. This is the power source, and you set a specific voltage, and you set a specific milliamps, okay? And then you allow the gel to run. So it will take some time, for example 40 minutes, 1 hour it will take for the whole thing to run. One more thing is that with the samples you should also add a dye. You should also add a dye. What is this dye used for? So when you are running the sample, you won't be able to see the samples, because you can't see the proteins in your own eyes without staining them. So that is why you use a dye. So the dye tracks the migration of the samples. So suppose you can see the dyes here, so which means your sample has migrated to this position, okay? So what we often do is we allow the dyes to get out of the gel, and then we stop the gel. So that's the role of the dye. It is used for tracking the migration of the samples in the while the gel is running. And then we dismantle the whole system. We take out the gels. We actually open those gel cassettes, the glass plates, and then we take out the gel and then we stain the gel. So what is the stain used here for staining the protein samples? Kumasi blue. Correct. Kumasi blue is used for staining the gel, and you have to stain the gel, you have to also destain the gel so that only the protein bands will now be visible. So this will form this kind of a gel. So here you can see these are your protein bands. All of these are protein bands, and this is the marker. According to the molecular weight of the marker, you will be able to identify like which is your protein of interest. Because if you know that your protein is suppose 50 kilodalton in weight, and according to the marker, so suppose according to the marker if this is 40 and this one is 50, and you will notice that your protein will be here, suppose. So which means that or the band which has formed here with respect to 40 or 50, that is your protein of interest, because you would know the molecular weight of your protein. So that is how the whole setup is done, whole SDS page is done. Now coming back to the question once more, the first option was, the question was which of these statements are or is true. First option was ethidium bromide is used to track the progress of the electrophoretic mobility. Here it is correct, but it is not correct with respect to SDS page. So be very careful, because this is given, this option is given here to trick you, because ethidium bromide is used in agarose gel electrophoresis only, not in SDS page. Okay, so this is a correct option but not correct with respect to the question. So therefore this is for DNA gel electrophoresis or agarose gel. So therefore this option is not correct. Next, coming to beta mercaptoethanol, this we have also seen that beta mercaptoethanol is used in SDS page so that the disulfide bonds can be reduced. So this is correct. Protein migrates towards the anode, it is also correct. It is a positive electrode. The protein is negatively charged, it will migrate towards the anode. And the last option is also not correct, because the lower molecular weight protein will migrate faster than the larger molecular weight protein. So that's all about this question. So the correct answers are B and C. Okay. So for more information, you can follow this particular NPTEL course, and the YouTube link is also provided here. And it has also discussed about native gel electrophoresis. So if you have any questions, you can go back to this NPTEL course and you can follow it. Okay.